Okay, a very good morning, everyone. I know it's really early and I really appreciate your attendance for this morning. Today we're just going to have a webinar session, more of like a sharing and discussion on the development trends and challenges in the commercializing CGT production. Okay, let me just next slide. Okay, this is actually the agenda for today's webinar. But before we go any further, we would like to do a poll of the audience just to as you can see right now, the poll just popped out. It's more of just to understand our audience role today. So you can just pick one that is most suitable for your current role and responsibility. Just sharing so we know where everyone is coming from. Yep. Okay. Okay. So we have a good mix here, as we can see. As I can see, we have people working in the industry, and there's people who are not currently involved in the industry, but they have some prior knowledge, and there are people who actually not familiar with CGT at all. So today, like I say, this session is more of like a sharing and discussion. So we're going to keep it open and hope that everybody will learn something and from this whole webinar. Okay, so we're just gonna continue on the agenda for today. We're gonna start off with something very light, which is just the overview of the CGT industry and what are the current market trends. After which we're gonna go to something a little bit heavier, which is the key challenges and the pain point that the CGT industry is facing, where we try to commercialize production. And after that, we're going to have a bit of discussion on what we call the CGT total solution. And after that, we're going to end it off with some future trends, development, and development opportunities. Okay, if any point, if I'm going too fast, maybe you can just ping me and let me know, and we can slow it down. Okay, so moving on, while I was preparing for this webinar, this article came out in Singapore News. It's about this little 3-month-old young Singapore baby in need of a gene therapy treatment. But as you can see, right, the cost of the treatment is actually 2.4 million. So the parents is actually having this fundraising campaign, hoping to appeal for donations to help this baby. So this gene therapy treatment that this baby Lucas is hoping to get is Novartis Zolgensma. It's actually a one-time gene therapy treatment for this very rare disease called the spinal muscular atrophy, which is short form as SMA. So how does this gene work is basically this Zolgensma will address the root cause of the disease by replacing the missing or the non-working SMN1 gene with a new working copy. And with the new copy, hopefully, the gene will then helps the baby in terms of a regular growth, because without this gene, there's the motor function issue. It's not working for this baby Lucas. So I think the most astonishing thing is the cost of this treatment. It's actually 2.4 million for a single dose, but good thing is he'll only need 1 dose for the treatment to work. Yeah. Okay, so this is just a sharing because this news came out and I thought it was very timely. Another one that is probably more familiar with people who has been in the industry is the Emily Whitehead story. She is actually the first pediatric patients to actually successfully receive the CAR T cell therapy treatment. Her story was a little bit unfortunate because typically children that is diagnosed with this type of leukemia, which is acute lymphoblastic leukemia, or short form as ALL, they actually has a recovery chance of 85 to 90%. However, it wasn't the case for her. She actually relapsed twice. So then the leukemia became resistant to the treatment, and her doctor basically sort of like gave up because they have tried all methods. So in the end, they actually recommended her a hospice care and expect her to just live for just a few more weeks. But of course, the parents doesn't give up. So they heard about this experimental treatment back then called the CAR T cell therapy, and it's in the phase 1 clinical trial. So in April 12th, they enroll her for the clinical trial, and she's the first patient to receive this CAR T cell therapy treatment. So how this therapy work is the process is quite straightforward. Is that they will collect some of her healthy T cells and they will genetically reprogram it so that the T cells will recognize the cancer cells, and then they will attack the cancer cells when they infuse the modified cells back into her blood. So great thing for her, as you can see, the treatment was highly successful, and she has been cancer free for many years now. So her cancer basically went into remission, and she has grown into a young adult. Okay. So that brings us back to the history of this CAR T treatment. It's not new per se, like it started way back in the 1989, where scientists start to create T cells that express this chimeric T receptor. But it wasn't until 1993 that we have the first generation, but it wasn't very effective. So for decades, right, we know that the foundation of cancer treatment has always been either you do a surgery to remove the tumor, do chemotherapy, radiation therapy, and this is even in fact up to now they are still the critical mainstay of treatment. But but in the recent year, there has been a new category of treatment, which we call them the immunotherapy. So what is immunotherapy? It's basically therapies that enlist the strength and the power of the patient's own immune system to attack tumor cells. So CAR T cell treatment is actually a form of immunotherapy. And of course, it has generated substantial excitement among researchers and of course, oncologists. Okay, so um, the first generation of CAR T, like I say, it wasn't very effective, as you can see right here. But there's a second generation that after 10 years of development, we came out with the second generation. The difference between the first and the second, as you can see right here, the first generation of CAR T basically just contain a single signaling, while second and then the third generations contains one or two additional co-stimulatory domains to enhance the survival as well as the proliferation and the persistence of the activated CAR T cells. And more recently, we have the fourth generation, which short form as the TRUC, because it is known as the newest approach in adoptive T cell therapy, where the CAR T cells are genetically modified with an inducible cytokine gene cassette. So this card, imagine it's like a vehicle, right? It actually brings in additional cytokine gene cassette, which will actually help to immediately secrete cytokines such as IL-12, interleukin 12, upon the CAR T cell activation. Okay, okay, and sorry, my mouse went missing. Okay. And after that, in 2017, that's where we have some groundbreaking scientific advancement. Is where we have 3 CAR T approval. Just in 2017, there's actually 3 since then. But all these CAR T approval are mainly for the ALL, which is the illness that we covered for Emily Whitehead, and also for DLBCL, which stands for diffuse large B-cell lymphoma, since 2017. So unfortunately, all these approved CAR T therapies are focused on treating selected relapse and refractory liquid tumor, and the percentage of these patients are actually made up less than 5% of. So as you can see, not a lot of so-called cancer patients can actually benefit from this. But like I say, this is just the start of this whole CAR-T, story. Okay, so let's to the next slides. Okay, so far since 2017, there has been 6 CAR-T cell therapies that has been approved by the FDA. All are approved for the treatment of blood cancer, including like lymphomas and some form of leukemia. And the most recent one is multiple myeloma. So as you can see, the pipeline of instigative CAR-T cell therapies has rapidly extended to more than 500 trials underway in 2019. While the earlier studies of the CAR-T usually target CD19, as you can see right here. So for the initial first few drugs, as you can see the timeline at the bottom here, they are targeting at CD19. But more recently, the latest drug that has been introduced and approved targeted at this antigen known as BCMA. Which it stands for B cell maturation antigen. It's also a common cell surface antigen in multiple myeloma, as well as a number of other cell surface targets. So the most recent one, which is approved in the United States in February 2022, which is last year, is Carvykti, which is an anti-cancer medication to treat multiple myeloma. Okay. Okay. Okay, so the CGT market overview, as you can see here, there's very exciting growth as the market is growing, that's for sure. So according to the research report and forecast from Frost and Switzerland, is that in the future 5 to 10 years, CGT market has a good growth trend with an annual growth ranging from 10% all the way to 30%. Okay. So when we talk about CGT, there is a particular nation that we need to, a lot of researcher will focus on is actually the China market. So that's why we have these slides. We talk about the Chinese CAR-T race. So as you can see right here, in fact, there's more CAR-T treatment and clinical trials in the China as compared to US. This is the reason for this phenomenon or this trend is because it's driven by the strong policy support that China has. So China has already become a hotbed for CGT development, ranking second in the world with more than 1,000 clinical trials either conducted or underway and thousands of related patents that was granted by the Chinese government just between 2 years, which is 2017 and 2019. So it is booming, right? And it's usually led by a mixture of not just local, but also foreign company. So foreign companies are also very interested to have their research done in China market, so which drive this boom. So the 3 key drivers for China's thriving CGT sectors include the first one is there's greater regulatory clarity now, and there's industrial regulation on quality standards. Number 2 is that the maturing infrastructure for the clinical study has actually increased that results in an increase in the global multi-center clinical studies. There's been collaborations between the local biotechs and multinational countries, such as Fosun and Kite, or Juno and WuXi. So they have such collaboration that is very powerful and strong, which is beneficial for the China market. And of course, a booming ecosystem with the government industry collaboration, driving basic biomedical research towards high innovative IPs in CGT. Okay. All right, so next, we're just going to compare CAR-T and other CGT development process against conventional drug discovery and development diagram. So elements that is not being utilized for T cell therapy are in the gray italic. Maybe the gray are not showing up in the slides, but they are the PKs and the heat leads in here. Okay, so very similar to conventional drug development, target identification is always the first step in gene to drug reverse pharmacology approach. A good target is, of course, crucial in delivering the highest drug efficacy and, of course, with the lowest incidence of adverse effect. Lead identification and optimizations. For a commercial drug, usually we look at the hit leads and then we select, we have a lead optimization and then we select the candidate drug. But for CAR-T, it's not the case. Mainly CAR-T starts by rational design of the CAR based on the domain function. So that's the main difference. That's why we don't have hit and leads for CAR-T drug development. And for the clinical trial phase, oh, no, we should talk about this first. Okay, so the preclinical biological testing, whether is it in vitro or in vivo, for commercial drug, we usually look at the PK and the DK. PK meaning the pharmacokinetics. But because for CGT, the drug itself is a form of living cells. That's why instead of PK, we are using CK, which stands for cellular kinetics, is adopted in this context. And also the clinical trial design for CAR-T treatment is actually different from commercial drug per se. In commercial drug, we have phase 0, which is mainly a test of like the proof of concept molecules. And then we have phase 1, which tests for the safety. Phase 2 is the efficacy of the drug. And from phase 3 onwards, it's confirming the safety and efficacy. And then after that is the post-market surveillance. So that is for the commercial type of drug. But for CAR-T, it's slightly different. The clinical phase lasted entire from phase 1 all the way to phase 3 and onwards. So that is the whole the whole phase, I would say, is actually clinical trials all the way. And during phase 1, they were generally, we have carefully selected a very small group of terminally ill patients where they have no other alternative treatment, but, probably, CAR-T is their final last hope. So after that, we will evaluate the primary objectives of the same thing, which is the safety and the feasibility of the treatment. There are secondary objectives, which could be a proof of concept studies. And in phase 2, the primary objectives is the efficacy of the drug. And how can we measure such efficacy is by measuring the overall remission rate, which is known as ORR. And as we proceed from phase 1 to phase 3 and beyond, there's increasing expectation for the product characterization. And of course, as you move to phase 2 onwards, we need to be in compliance with CGMP. And then from phase 3, besides, again, product characterizations, assuring product quality, and of course, most importantly, is the consistency in manufacturing. Okay, so now we're going to talk about the key challenges of CGT commercial production. This is the patient journey from start to finish. There's 3 key points that we will cover. The first one is, of course, the complex manufacturing and the supply chain. Next is from start to finish, it requires a high-touch commercial model. It needs a lot of manpower that's involved. And of course, with that, it comes with the costs and other a lot of training requirement to make sure that all the personnel that is involved are highly qualified. And last but not least, to even enter for the trial, the patients need to either be able to pay and to be able to be considered for this CGT treatment. Okay, let's start with the manufacturing process. All the CAR T treatment in the market out there, majority of them, I would say about 75% are autologous, which means one batch, one patient. And because of that, it is complex and it's costly to manufacture. Supply chain is also very tricky. And the manufacturing of this autologous CAR T today is largely centralized with limited economy of scale. So think of it like modular clean room and lab-like environment as compared to large manufacturing plants. And because of these, there's usually a shortage of manufacturing slots, either for the CAR T cells or even the requisite, which is the viral vector. These two are often cited as the key bottleneck. So the viral vector, as you can see right here in the diagram, the production for the viral vector and the plasmid, these two are the very important starting material. It's very complex and very long. So the viral production relies on the vector production by stable cell line. In this case, it can be a retroviral vector or by transient cell line transfection for lentivirus. And then they have to undergo various steps. As you can see, there's concentration, purification, formulations, etc. And then for the plasmid production, which usually initiated by E. coli master bank. And then after which we have plasmid replication, which is promoted by fermentation. After that, there will be cell lysis. And then there's pre-purification steps like filtration, precipitation, solid liquid separation, etc. So as you can see, it's really complex. And to perform such process, you need people that are highly trained. And because of this, there's a demand for talent in this area. And as more and more people try to get into the CGT market, the demand for this talent is even higher. And then it pits company against each other as they fight to attract this individual with the appropriate skills. So there's the lag right there. Okay, so besides the manpower issue, what can we do to sort of like help with the manufacturing process? So one of the way is by this holistic approach of the cell therapy platform by using the space isolation system. So before I go into more of the details, I'm just going to quickly show you a quick animation on the next slide so you have a better understanding of this space isolation system. Okay. So let me just play the video. It's just a very quick one. Okay, so in this video, actually, you see two equipment. One of them is the isolator, which we call them the cell prep station right here, as you can see. It basically provides a dynamic Class A laminar flow system. So it's a Class A environment inside the isolator itself. And it can actually be it's actually sterilized using this VHP, which stands for vaporized hydrogen peroxide sterilizations. So this process can be automated in a way that will differ from like traditional, you spray with the alcohol and then IPA and then you have to clean it. And then also, depending on the need and your requirement, you can have different embedded modular combination right here, like the one that you see in the video. You can have robotic arms, you can have a refrigerated centrifuge, there's a docking system, microscope, all sorts of EM monitoring, waste, video surveillance, etc. Okay, and of course, the other equipment that you see in is actually the honeycomb culture system. It's actually a closed system using this RTP valve system, which stands for the rapid pot docking. So just now you see the little robot that went into to pick up one of the pots and then did the attachment to the cell processing station. So this rapid docking technology of the RTP honeycomb incubator, this whole docking process is completely sealed. So you don't need to wait for any additional sterilization, and the door will automatically open. So all these will reduce manpower intervene, and this will ensure internal sterile environment. The transfer time is actually less than a minute. And then also there's a security lock mechanism to ensure that there is no possibility of leakage until the valve is fully sealed. Likewise, this incubators also uses the VHP sterilization. And so same, this process require less time than other form of sterilizations. Again, all these can be fully automated, and the honeycomb incubator also communicate with the honeycomb cultures directly via Wi-Fi communication. So this can ensure the integrity and traceability of the data in the process of the cell culture. So anything will be traced and caught in the audit trail. So you can always refer to that for if you need to check anything, like auditor needs to check who has access to the cell line and etc. Okay. So if you were to compare the isolation system versus a typical grade B and A, it is, of course, it's a fully closed system, unlike the grade B and A, where it's open system. The person that's working has to have direct access to critical operating areas. Of course, with a lower grade, like in the background, the cost for the startup and the maintenance cost is, of course, much lower as compared to B and A. And also, some other more other challenges that face in B and A is like the gowning. So the gowning can be reduced. And like I said, the sterilization wise, this is an automatic one as compared to manual. And even because of that, the downtime that you need for cleaning is much lesser as compared to regular ones. Yep. Okay. If the isolation is not something that you would like to consider, you can always consider using a single-use system. So this is similar to the isolation, but instead it's using a single-use system, as you can see right here. The cell process, the CAR-T process has many, again, it's a long process. It is time consuming, as you can see here. And at every step, there is almost pain points. So what we can do as equipment maker here and to help is we can come up with equipments that can simplify this process. Okay, so what we have here is a single-use cell processor. Basically, this single-use cell system allows, it's a fully closed system. It's automated, and it can do multiple process with just one equipment, basically such as the cell separation, concentration washing, and cell preparation. So by using this, the respective single-use consumables and the software and the equipment, you can one equipment with the different consumables attachment, you can perform all these steps. So what's the evolution of the CAR-T cell manufacturing process is what we can see is that we are generally moving from open system to a functionally closed system, because open systems are, of course, more convenient. They're easy to adapt because CAR-T cells generally we start with adherent cells. So that's why open systems like with the T cell, the T flask growth, all this is generally easier to implement. And also, but then of course we are looking at moving them to closed system, like such as using single use. But then of course, single use, for those who have used them, you know that single use requires skilled operators at all stage, like be it the installation of the bags or the consumables and things like that. Also, one of the other way is that you can see that we are replacing manual processing with automation, especially if you want to scale up to commercial scale. End-to-end automation is of course desirable, but then it will require very precise planning for the execution of the component in the manufacturing chain. A failure of one device can actually result in significant impact on the production flow. But of course, with robots, ideally you can reduce the human error and variability. So like a robotic arm system, it can be incorporated like the one we see in the video. So efforts has been made to automate these culture systems to improve their scalability. However, automation also come at the cost of flexibility. Any minor adjustment in the process often require a major change in both the hardware and software. Another evolution that we are seeing right now is that, like we have mentioned, current treatment are all autologous, which means is one-to-one. But hopefully we can move away from this and we can do to allogenic, which means that we can have a donor, and then eventually it can goes off to the general population, to the general population. So think of it more like off-the-shelf treatment by using donor cells to create a standardized target cell therapy for the general population instead of the highly individualized batch. And of course, with this, you have a bigger market, and maybe the cost and all other we can lower it down. So the pros of using an autologous, which is the 1:1, of course, it is safe and it has low CRS, which means cytokine release syndrome, which will result in lesser side effect. But the cons is, of course, it is a low manufacturing capacity. It has a long lead time. As for the allogenic, the cons are, of course, you have to think of the safety concerns, like, donor and receiver, they may reject because it doesn't come from the patient itself. And usually, as we know, these patients are very sick. Any other adverse effect might be deadly for them. So, and also, by far, there's no commercialized product yet. But if this is successful, it can have high manufacturing capacity off-the-shelf product, which means that the cost will generally come down. Okay. Oops, it's not. Okay. Logistical issue is also one of the main reasons why the manufacturer is facing, is that the logistic for manufacturing and delivering CGTs are always hard. Let's say autologous cells, for example, the journey, it's long, and usually it's not at the site where we manufacture. So what happened is that the cells will be extracted at a clinic or hospital, we call it ephesis, and then after that it's being dispatched to specialized lab, like the centralized lab, where the treatment is then manufactured, after which it will have to be sent back to the hospital where it will be administered to the patients. So all this is very time pressure and there's strict quality control. Usually the supply chain is a cold supply chain to maintain the temperature. So all these actually has many potential source of error. And so this whole process, which is slow, and as you know, like I already said, the patients are always in the late stage of the disease. Any delay can actually cause can actually disrupt the whole entire supply chain flow. Okay. So what manufacturers can do is that since the process is so complicated, they can actually work. They can have long-term contracts with global logistic major and specialize in medical only couriers to optimize their supply chain and to ensure handling quality. Some also can partner with software companies to offer digitalized technology solution that tracks and trace and track the cell therapies with enhanced transparency. So whatever it is, just think of it as all the things that you are doing, you are trying to do a risk mitigation. So other risk mitigation are such as planning the supply ahead and like inventory planning, because the most recent COVID-19 crisis is a good lesson for all of us, is that how sensitive the supply chain can be if, let's say, we have a COVID-19 crisis where there is such a severe supply chain disruption because of all the travel restriction. So the most important is that we want to reduce the vein to vein time between the clinic and then to the manufacturing site and then back to the clinic again. Another way is that we do decentralizing. So if you look at it, think of it like dialysis treatment, right? So instead of having the patient going to the hospital and then staying there throughout the entire whole treatment, we can have outpatient centers. If you're not familiar, another way is you can think of the Ray Kroc story. If you don't know who Ray Kroc is, he's actually the man who franchised McDonald's. So, instead of going only to maybe the United States to eat French fries, he actually franchised it out to all the various countries, the various different places, so everybody can eat McDonald's, can have the famous French fries. But of course, when you decentralize manufacturing process, there are some concerns over this, is where how can you ensure that the quality is the same in all the different franchise, just like how can you ensure that you can sorry you can sorry my mouse okay so how can you ensure the quality and the safety and the efficacy of the product is the same, and it doesn't vary from site to site? So by decentralizing, it actually aims to reduce the delivery time. You can have them set up close to the hospital or even in the hospital itself. Think of modular manufacturing facilities like a GMP in a box solution. So all this has, especially this modular facilities, has actually driven a lot of significant interest in this area. It's also low cost to start up, which is also something that will attract manufacturers. Okay, so the aim of this platform is to decrease the manufacturing cost, simplify the logistic, and reduce the footprint. Most importantly, like I say, is to minimize the vein-to-vein time. And if you can do away with the cumbersome supply chain, of course, that can bring down the costs as well. But like I say, decentralizing will bring out other challenges like the quality management and the variation across sites. So that's something that manufacturer has to work on before this can happen. So the high cost and affordability issue is something that is on everybody's mind, right? Like, we know that it's labor intensive, it has a complex supply chain. So of course, the cost of goods sold remains high. And of course, the modalities like the starting materials, like I say, the viral vector, all this, they still suffer from capacity constraint. It has long lead time, and there is significant upfront investment that is required for this viral vector production. The reason why it's expensive is also because it's low yield due to the low transfection efficiency, and the use of adherent cells really is one of the limiting factor. And also CGT concentrate into a short course treatment, like the Zolgensma that we see is actually, in fact, a single treatment, thus making it very expensive for payers, And if you compare to the traditional payment mode where payers have to make a fixed upfront payment, the payer actually have to bear all the risks of the new therapies. What if the therapy doesn't work? But I already made all the full payment, and then maybe I didn't live long enough to see it work, or I died, or it just doesn't work for me. So there's a risk involved, and the payer might not be willing to do so. So what a lot of payers and the biopharmaceutical companies are coming out is that they're coming up with new paying methods and making it more affordable, such as they have this contract basis whereby there's a share risk. So what we can do is basically they can outcome based, like if you hit certain milestone, then you pay certain percentage. So this way, the risk is being shared by the payer and the manufacturer themselves. And of course, with the government funding and support, that will definitely help. And maybe insurance company can come up with a more sophisticated health plan to explore options that include funding of the gene therapy as well. Like I say again, the inpatient to outpatient setting, if you can have like the dialysis center, of course, that will, help to reduce the cost, whereby you don't have to have the patient staying in the hospital throughout the whole time. And, you can have outpatient centers where they will go in and receive treatment. But of course, with the outpatient center, it doesn't mean that you don't need the hospital per se. It just partner with the hospital. So that will definitely help to reduce the cost by just staying in the hospital throughout the whole entire process. Okay, and of course, there is increasing regulatory requirements. Uh, this has CGT is like any other drug. It needs to be compiled with CGMP, and of course with any other laws and regulations, as you can see right here. So there's actually FDA also have this gene therapy CMC guidance that has been available. So manufacturer can refer to that when they develop the therapy itself. As more CGT are being developed, the potential of adverse effect and we starts to have more knowledge of them. And of course, regulatory body like FDA are also putting pressure on CGT manufacturer to adhere to the new standards. Quality control for quality attributes such as the identity, purity, potency, and the safety is becoming increasingly difficult as there's more parameters to assess. And then also all these assays that needs to be conducted are usually expensive, and they can be quite long. It takes from days to weeks to do because they are generally just cell-based assays. And also the regulatory requires all these assays to be validated, and then that's just add on to the pressure that's out there. So what FDA is doing is that actually FDA renamed the Office of Tissue and Advanced Therapies as the Office of Therapeutic Products. This is a new entity that is basically has growing. It also growing its capacity to to handle the growing number of the application that is coming in. This is according to the FDA itself. And meanwhile, FDA is also expanding its reviewing capacity of the increasing number of cell and gene therapies in the pipeline, which is another positive development for the field. And regulatory agencies are taking steps, like basically they hire more people so that they could sort of like, increase their capacity issue so that they won't delay the timeline in terms of the approval. And ideally, we hope that there will be sort of like a draft guidance to all the cell and gene therapies, so it will help to continue to path a clearer way to the approval for all these manufacturers. Okay, so we're coming to the last two slides, which is looking ahead what kind of challenges or what kind of development we can look at. So solid tumors are increasing in the focus of development activities. Traditionally, cell and gene therapy, like I say, has been for hematologic cancer, which is the liquid cancers like blood cancers, etc. So solid tumors now made up 45% of the total oncology trial. And so that's very exciting right here. And also, obviously, like I say, the most important raw ingredient is to manufacture the viral vectors. So CDMOs have also tried to expand their capacity in the recent year, but of course, they have yet to meet the demand. Because viral vector is not only required for just CAR T, right? As you can see, even like the COVID-19, the mRNA, they requires viral vector as well. So it's a very competitive market for these companies in both the short term and the long term. So companies that lacks access to this viral vector can have delay in their clinical trials. The wait time is about 16 months in 2018, and they have grown as high as 3 years in more recent. Even worse for established commercialized product, a break in the supply continuity in the viral vector can prevent the patient from accessing these medicine. Like I say, back then when we had COVID, maybe most of the viral vector has been redirected to other form of treatment. Uh, can we think of other ways instead of using viral vector? Yes, there are other ways. As we know that currently, the current vectors derive from AAV cells, which is adeno-associated virus or lentivirus. These are the most prevalent in this industry. AAV is the leading viral vector for gene therapy, whereby lentivirus is the primary vector for cell therapy product. So we can use other ways is, as we know that currently the viral vector uses adherent cells, which is limited by the size and the capacity. So what about trying to change them to suspension cell line? Then that will definitely allow us to grow them in large tanks, and that will help to reduce, in terms of the capacity issue. However, we can't say, okay, tomorrow I'm going to use suspension cell line. Some of the manufacturers are already, working towards commercializing. It's too late for them to change the cell line or to adapt to the latest optimized viral vector technology. So transitioning from adherence to suspension base is something that we'll work towards too, but then it will come with other regulatory hurdles that we need to overcome. And of course, there are non-viral vector methods that includes physical and chemical strategies, such as lipid-based nucleic acid delivery system, naked plasmid, electroporation, sonoporation, etc. But all these are all in the testing phase. Okay, oops, sorry. Okay. And ideally, hopefully we can have a harmonized production protocol, like similar to what we have for MAP, like MAP production right now is pretty much standardized, it's broadly accepted. So if we can have such standardization for CAR T cell manufacturing, that will definitely enable a more rapid assessment by the regulatory agents and then facilitate the commercializing to meet the increased demand. And last but not least, of course, we will have, hopefully we can develop more allogenic product. So with these, again, advantage in the manufacturing includes the reduce of the cost of goods, a simplified supply chain, and it will helps with the long vein-to-vein time and things like that. So this is something that is a lot of researchers are hoping to move towards too. And ideally, this might be what in the future will be. Okay, so in summary, there is no one size that fits all solution, as you can see. If I choose this method, there might be other challenges that I need to overcome. So what we see right here is ideally as we progress, we're going to bring the cost and of course the risk down, and then of course, it can reach to more patients out there. So think of closed system, automation, and digitalization. And instead of scaling up, we can think of scale out, where transit from clinical to commercial. We can decentralize. So it's more of like the combination of various factors and how we work towards this. Yep. Okay, so sorry, this is the end of my presentation. So this is the Q&A session. Thank you so much for your attention for the webinar. For the Q&A session, actually, I also like to invite one of my colleagues. His name is Mr. Vivek Zhao. He is actually the process development expert in the team, and he is the expert on CGT market. So, yeah. Thank you all. So we can take the questions from the floor. I know I have overran the session. So yeah, Linda and
Yes.
Mr. Vivid. Yeah, hi. Sorry. Yes.
Hello.
Thank you.
This
Thank
is
you
Vivid.
very much, Claire. Hi, Vivid.
Thank you.
So
Yep.
please do post your questions on the chat, or you're very welcome to unmute yourselves and ask Claire and Vivid your questions. Thank you very much for that presentation, Claire.
Yeah.
I'm sure there must be some questions out there. Don't be shy, everyone. We've got plenty of time, I think, to run through any queries, or even if you have comments, suggestions, you're very welcome to share.
Yeah, like I say at the start, this is more of like a discussion and sharing session. So if you have any idea, I'm sure that the people on the floor in the audience would love to hear about it, and it's open for discussion.
Yeah. So from the very basic to the very technical question, please let Claire and Vivid have a go at answering. Just so everyone knows, this session has been recorded and it will be posted on ISPE Singapore's YouTube page, along with all the other webinars and Technical Tuesdays and other recorded videos that we have there. You will also be able to ask any questions after this session. If you'd like to just let us know, we will forward it on to the team at Tofflon so that they can address your queries directly. So is someone brave enough to give us a question?
Perhaps it's too early, or everyone is still trying to ingest all the
That's right.
Try to take in all the information. I know there's a lot of things to consider in the CGT market, like the challenges that everyone is facing. So,
and typically, Claire, I mean,
yes,
these kinds of forum, what would one of the key issues be that people ask about? What's the common concern?
Well, usually when it comes to any new modality, people is very concerned with the regulatory requirements. There is no standard guide per se now, because the process differs from every treatment, right? It's so individualized. So it's very hard for FDA to say, okay, everybody must follow this set of guidelines, because it's just difficult, right, at this point right now. So ideally, they can just, we are sort of like learning as we go. And one thing is that we can't really assess the long-term effect of the CGT because it's so new, right? Like the first treatment is 2017, and you can't say that 10 years or 20 years down the road, there won't be any adverse effect. Nobody knows because we are not there yet. So like I say, all these are just a lot of uncertainty that's out there. So it's more of like moving forward with good faith, and hopefully they will all goes in, like the therapy will work for them, and there's no there will be no side effects in the long term. So things like that. So I think regulatory, definitely the FDA are working very closely with all these CGT manufacturer, collecting all the data, consolidating them, and then sharing it out. And hopefully we can come with a standard guide that, we can sort of like, adhere to. So this will definitely make CGT more, so like the whole process better in that way. Yeah.
Thank you. And Vivid, do you have any comments?
Yes, and from my side, I think different that was decided and concerned about the pieces of the subjects from the certain countries. Like recently, we have a technical transfer to Russia, and they have only concern about 100 patients per year, and depend with certain patients and the different process and equipment they will select. That's from my side.
Thank you. And so we still don't have any questions. So I guess maybe you've covered everything, Claire. Or certainly, we might get some emails with regards to inquiry directly. So if there are no other questions, then I think we will end this session.
Yep.
And if you have any last thoughts, Claire, to share with the team?
So like I say, this CGT market is really exciting and new. In the upcoming years, we definitely will see a lot of new trends coming up. So hopefully that will become the next pillar in cancer treatment. And yeah, so since there's no question, if anything, my email is up there. You can always drop the question to me, and we can take it from there. So once again, thank you everyone for attending this morning webinar. You have been a very great audience. So yep, thank you and have a great week ahead.
Thank you, Claire.